Can we share a screen? Can you show your screen?
Mm-hmm.
Okay, cool. It's working. All right, so my great pleasure to introduce Ilya Levental today. And so Ilya is one of the most innovative and creative scientists working in the field of membrane biology. And so I wasn't surprised that many people here asked to meet with him, and I think he's having very good series of meetings. His training was initially in bioengineering. He got a Ph.D. degree from University of Pennsylvania in 2008. Then he went to work with very famous scientist Kai Simmons in Germany, in Max Planck Institute of Cell Biology. So he was a Humboldt fellow there from 2008 until 2012. From 2012, he got his own faculty position as an assistant professor in University of Texas Health Science Center in Houston, where he established his lab and published, series of papers on membrane biology, which are very interesting. And then this year in 2020, in the middle of the pandemic, I guess, he managed to move to University of Virginia. I didn't know it was possible to move during this time.
It wasn't easy.
It wasn't easy.
It wasn't easy.
So, and now from 2020, from this last year, he basically associate professor in the Department of Molecular Physiology and Biological Physics in University of Virginia. So really looking forward to this talk.
Thanks so much. Thanks to the original Ilya for the invite. Thanks to all of you for making the time. It's a real pleasure and an honor to be invited to this fantastic department. So I'm really excited to share our stuff. And I'll get right into it because I got a lot to tell you. So based on the talks I've had already and reading through websites, I don't think I need to tell you that membranes are amazing. I think you all agree with that, at least from the physiology and cell biology side. And I'm here to tell you maybe a little bit that how amazing they are from the materials and biophysics side. So as you all know, on the molecular level, biological membranes are essentially quasi two-dimensional fluids, so hardly any stable interactions in the plane of the membrane between any of the components. But at larger length scales, the same materials behave as robust, elastic sheets capable of sustaining large tensions and deformations, as you're seeing here. There's an isolated plasma membrane from a mammalian cell being yanked and stretched by various torture implements. So very much not like a fluid and much like a solid. But then zooming back into the molecular scale, some of the chemical and physical gradients that are inherent to biological membranes are practically unmatched elsewhere in nature. So if you think about the chemical environment transitioning from completely aqueous to completely apolar in the length span of just under 10 angstroms probably. And the same is true of physical forces that go from hundreds of bars of tension at the head group level to hundreds of bars of pressure in the hydrophobic core, again, within less than a nanometer sometimes. So all of that stuff makes membranes really beautiful objects for study. But not only are they kind of interesting and curious, they're of course also very important biologically. And I think that's most easily illustrated by pointing out the fact that approximately a third of the mammalian proteome codes for transmembrane proteins, those that are stuck in membranes forever throughout their lifetime. And that doesn't include, of course, the many, many proteins that interact with membranes less stably than forever by protein lipid binding, by post-translational lipid modification, and interaction with other membrane embedded or bound proteins. And so I think in a mammalian context, certainly it wouldn't be unfair to estimate that approximately half or so of all bioactivity occurs right at or very near a membrane. But these membranes aren't just the host for all of this essential bioactivity. In many ways, they're also its guide. And I think that that's most easily illustrated by pointing out that in almost any context where it's been carefully studied, the composition and collective properties of the membrane matrix can have important consequences on protein structure and function. So from a receptor ligand binding to transporter activity, various membrane channels, other transporters, membrane embedded enzymes, and really just most broadly, oligomerization and conformation of membrane proteins have been shown to be regulated by composition and physical properties of the membrane that's solvating them. But there isn't really a kind of an overarching context that can explain this, but we know that the membrane is an important determinant of protein function. And at the same time, we've come to realize that mammalian membranes are much more complex and more diverse and more plastic than I think was appreciated until the development of this membrane lipidomics technology. So this is basically mass spec, but mass spec that's sort of ideally suited for identifying and quantifying all of the lipid species in a complicated sample like a mammalian membrane. And through these kind of studies, we and many others have found that mammalian membranes are composed of sometimes 1,000 distinct lipid species, all with kind of interesting dynamics in terms of how they're upregulated or downregulated in various cellular states. Just kind of as an example, in a study published a few years back, we measured how the plasma membrane lipidome changes as mesenchymal stem cells differentiate into various terminal and functionally different lineages in vitro. So this was an in vitro study, isolated MSCs, differentiated into osteoblasts versus adipocytes. And then we isolated plasma membranes and saw that the composition of the plasma membrane in these three cell types were clearly distinct. And in particular, osteoblasts from 3 human donors, that is differentiated osteoblasts from MSCs, acquired a unique plasma membrane lipid signature that was, sort of really specific to this particular lineage compared to its precursor cells even. And these lipid signatures could be manipulated in a way that I'll show you in just a second, but either genetically or biochemically, we can manipulate these lipids in order to actually tune how efficiently MSCs differentiate down these various lineages. So these are not just readouts of differentiation, they're also in some way an important guide for the differentiation process. And of course, if these 3 very closely related cell types were distinct with respect to their lipid composition, if you look between tissue types, some of the differences become really quite dramatic. So this is sort of comparing 3 representative cell lines of 3 different tissue types. And we see that both in the head group level, but perhaps even more dramatically in the hydrophobic acyl chains of the lipids, we see quite major differences between these 3 types. And even here, what we're calling the neuronal lipid composition isn't something that's set. Not all neurons are the same. And even those neurons change through various physiological processes here, for example, development. So these are neuronal synapses isolated from postnatal rats over the first 2 months of development. And we can see, again, both in the head group level and in the acyl chains, some really striking changes over this kind of early neuronal development. And then I mentioned this point about plasticity. We've been investigating this sort of notion that membrane composition can very easily be manipulated by things like dietary inputs, because for fats, it's actually literally true that you are what you eat. It's not true for proteins or DNA, obviously, but the fats that we eat are indeed incorporated in toto from dietary sources into membrane lipids. And so this is a kind of ridiculous cartoon to demonstrate that, but here's some data. Both in vitro and in vivo, we can feed a polyunsaturated fatty acid like this omega-3 DHA. It's the major component of fish oil. We can supplement it into culture media, or we can supplement it into animal diets. And this exogenous fatty acid that mammalian cells don't make and that mammals themselves typically don't have gets incorporated to really striking levels where up to 30% or more of all phospholipids in murine heart will contain this exogenous fatty acid as one of their tails, like so. So I think a really, to encompass the challenge that presents our lab and I think the field in general of membrane biology is to understand these mechanistic connections between membrane compositions and how they change during various processes, and then the biophysical properties that result from those compositions, and ultimately how those feed back onto the functional phenotypes of the membranes and membrane proteins in the cell that we care about. And that's sort of how we structure our lab into these interconnections between these things. And within that broad context, I hope to tell you two quick stories today. And the first one being how the organization of the plasma membrane and endosomal membranes regulates trafficking, subcellular trafficking. And to do that, I like to start off with something that I call a membrane mystery. So it looks like a really trivial result that hides a really weird observation. So this is an image of the subcellular distribution of a transmembrane protein called LAT. It's an immune system protein. It's an adapter, single transmembrane alpha helix, and it lives and works at the plasma membrane, and here it is mostly at the plasma membrane in this generic mammalian cell. So now this is the subcellular distribution of a truncation of this same protein. A whole bunch of the protein is cut off, and as you can see, it still traffics nicely to the plasma membrane. And here's this mutant. This is a point mutation with 2 amino acids replacing 2 other amino acids in this protein. So this doesn't look like such a big mystery, right? Seems like a pretty trivial result. There's a whole zoo of trafficking determinants in various proteins, and this protein has one. This truncation doesn't affect whatever trafficking determinant there is, and this mutation breaks it. And that's basically exactly correct, except that this truncation leaves nothing of this protein but the hydrophobic, single hydrophobic transmembrane alpha helix. This is just 20 amino acids, all of which basically reside in the membrane. And this mutant, which completely breaks plasma membrane localization, is a point mutation of 2 hydrophobic amino acids, 2 alanines in this transmembrane domain, mutated to leucines. So hydrophobic amino acid to another hydrophobic amino acid makes a huge change in the trafficking of this protein in total. So this is the mystery that I'm hoping to resolve for you today. It's a kind of a really amazing observation every time I reflect on it. And to get into it, I have to take a bit of a long and possibly boring detour into lipid rafts. So probably many of you are familiar with this concept of lipid rafts. Many of you thought about it and probably been annoyed by it. But the idea is not so complicated and not so annoying. It basically, the thesis of the lipid raft hypothesis is essentially that lipids can self-organize to form structures in the plane of the membrane. And this is basically a fact. We know this is true because we know that certain lipids preferentially interact with one another. So lipids with long and saturated hydrophobic acyl chains will interact preferentially with cholesterol. Also, glycosylated lipids tend to interact preferentially with both of those kinds. And what I mean by preferentially is that they would much rather interact with one another than with highly floppy, unsaturated tail lipids. And while the kind of binary interactions are really quite weak, right? There's not that much difference between this interaction and this interaction. In the membrane, all of those things behave as collectives. There's many lipids all interacting with one another, and these small binary differences can add up to produce large-scale structures that have been observed in a variety of model systems, usually relying on synthetic lipids and pure lipid model systems, both computational and experimental. We see this large-scale structures. These domains can be tens of microns large, and both of them behave as fluids. So to produce this kind of liquid-liquid phase separation in the plane of the membrane, where one of the phases has lipids that are these preferentially interacting ones. The sterols, saturated lipids, glycolipids all exclude these more floppy unsaturated lipids, which make the more disordered phase. So this is something that's been studied for a very long time in the biophysics literature, but it was never quite clear what the connection was between these simple models and real living plasma membranes, which are different in many, many ways. So the whole point of building a raft for a cell is to sort and organize proteins in a membrane. Right? So the idea would be that certain proteins could be included and concentrated in a membrane domain. Perhaps negative regulators of a given reaction could be excluded, and thereby signaling could be modulated simply by organizing proteins in the plane of the membrane. Or even the structure and function of a given protein could be affected by the surrounding lipids. But I'm sort of being intentionally vague here with a lot of coulds and mays, because while the function of membrane domains is really inherently based on their selectivity for certain proteins, we really don't have answers to fundamental questions, like which proteins go towards the more ordered domain, and why would they do that? What is the difference between a protein that prefers to be in this region versus this region of the membrane, and what is the ultimate effect of sorting proteins in this way? And the reason that we don't have answers to these fundamental questions is because the models have been suboptimal in terms of their complexity and physiological relevance, and the methods for isolating and analyzing rafts and ordered domains in cells have also been a bit ambiguous, to be kind. And within that context, I think the development of this model system has been a really breakthrough for certainly for studying ordered domains in biological membranes. So I'm just going to quickly introduce this system to you. We call these things GPMVs, giant plasma membrane vesicles. And what you're seeing here in this movie is some generic mammalian cell that's been poisoned with maleimide. And when one does this in kind of typical extracellular buffer, we see that the plasma membrane of these cells inflates into these large balloons, up to 10 microns in size, so really kind of quite big. And then after about an hour or so, these bubbles of plasma membrane will basically pinch off the cell and float up into the supernatant, where they can be gathered up for biochemistry or imaging or whatever what one might want to do. So biochemically, these are really a beautiful prep for a mammalian plasma membrane. These bubbles are completely clean of ER, basically any other internal organelle. But from the biophysical perspective, what's most exciting is that these isolated plasma membranes that contain at least a fair sampling of the lipid complexity and the protein complexity and abundance of a mammalian cell still phase separate into coexisting liquid ordered and liquid disordered phases, exactly as the synthetic model membranes would. So if nothing else, this is a really essential demonstration that this liquid ordered, liquid disordered business has something to do with what happens in living cells. But perhaps more importantly, methodologically, the system allows us to answer important questions like those ones I posed on the previous slide about which proteins partition between these phases. And the way that we do this experiment is very simple. I'll show it on the next slide. So when we started these investigations to ask these questions, what are the determinants by which that determine whether a protein partitions to the ordered or disordered phase, almost nothing was known. Which wasn't the impression that one would get by looking at famous textbooks, where you could find cartoons like this that had sort of very suggestive ideas planted in them. For example, that because it's known that an ordered membrane domain is relatively thick compared to a disordered membrane domain, because the lipids there are stretched out because of the cholesterol in there. Therefore, proteins with long transmembrane alpha helices would fit better into this thicker membrane environment, and that could be a mechanism for sorting proteins laterally. Now, unfortunately, despite the fact that this was in textbooks, there was never any evidence for this notion. And so we set out to test it. So the first thing we did is, actually this, I'm not telling it in the right temporal order, this we did much later, but using cryo-EM, it's possible to directly image bilayer thickness in synthetic membranes or in isolated biological membranes, and we're also hoping in kind of in situ, in previously living, now frozen cells. But yeah, so you can see that this thickness difference is clear and it's robust, and approximately 10 or 8 to 10 angstroms in difference in thickness between a more ordered and the disordered phase. So just pretty images showing that. But to test whether proteins care about this thickness difference, we constructed a series of model proteins. It's really transmembrane peptides. So this is the transmembrane domain from that protein LAT that I showed you earlier on. It has this kind of relatively long transmembrane alpha helix. And then we replaced the entire business end of this protein with this fluorescent reporter of RFP. And then to test, for example, this hypothesis that transmembrane domain length is important for partitioning, we just make a variety of mutants here by removing a kind of a 3 or even a 6 amino acid chunk from the middle of the transmembrane domain, or some kind of neutral mutation, or a variety of things that I'll show you in a minute. And then this is what the experiment looks like. So this is one of these isolated giant plasma membrane vesicles, and it was isolated from a cell that's expressing this construct. So the cell does all the hard work of making this protein, trafficking it to the plasma membrane. Then we isolate these bubbles, these giant plasma membrane vesicles, and we can stain them, for example, here with a lipid that prefers the more disordered phase. And we can ask, well, which phase does the peptide prefer, right, this transmembrane protein? And in this case, as I hope you can see, the partitioning is approximately equal between these two coexisting phases. Maybe a slight enrichment, which we can quantify very simply by comparing the intensity at this point versus this point. And that gives us the equilibrium partition coefficient to the ordered membrane phase, or quote unquote raft. And from that, you can get free energies if one is so inclined. So now, an example experiment shows that while the full length wild type transmembrane domain of LAT partitions this way, a deletion, a truncation of the transmembrane domain, which is now 6 amino acids shorter, pretty clearly enriches in the more disordered non-raft region of these phase-separated plasma membranes. Whereas this neutral mutation, where those same amino acids, those same 6 amino acids, are simply replaced by alanines, behaves basically like the original protein. And so we can do that for some 24 constructs that we created here, basically removing amino acids from different parts of the transmembrane domain. And we see that there's a very clear correlation between the length of the TMD and how well it partitions to ordered domains, which is very much in line with that simple prediction from the Alberts textbook. The longer the TMD is, the better it partitions to ordered domains. And this line here, this dotted line, is a fit to a very simple model, which basically posits an energetic penalty. In the most obvious way, it's a hydrophobic mismatch penalty. If the hydrophobic region of a protein and the hydrophobic region of the lipid bilayer don't fit, there's a cost to deforming the membrane. And so if you have coexisting domains, proteins will sort to minimize that cost. And we get pretty good agreement between this pretty simple model with our observations. So it turns out that you don't have to throw out your textbooks. That cartoon is legitimate. Longer TMDs do indeed sort to ordered phases preferentially. But equally obvious was that transmembrane domain length definitely didn't explain all the partitioning that we could observe. And here's the simplest demonstration of that. Again, this is the wild type transmembrane domain from LAT. And now we made a construct that was basically the same length, at least nominally the same number of amino acids in the transmembrane domain, but now the entire TMD is composed of leucines. So the length is maintained, but all the chemical information in the TMD is lost. And we can clearly see that these two constructs don't partition equally. This all leucine construct became a completely non-raft protein. So then that led us to hypothesize that there must be something in this TMD that's sort of acting like a code for preferentially recruiting these transmembrane peptides to the ordered domain. And what that code was, we basically hypothesized in two different ways. Either it's something like a motif, right? A specific sequence that binds a lipid or binds another protein, and you know, yeah, very, very sequence specific code. Or it's something, some generalized comprehensive physical property of the transmembrane domain. And to distinguish between these two possibilities, we basically took the same sequence but randomized it, right? So now it would have the same length and the same other physical chemical properties as the wild type protein, but now all of the sequence information is gone. And to our surprise, this scrambled up transmembrane domain behaved basically at parity with the wild type lat transmembrane domain, sort of confirming that it was some aggregate physical chemical property of the side chains in this alpha helical transmembrane domain. And I'll make a very long story short and tell you that I think we figured out what that aggregate physical property was, and it's basically the size of a transmembrane domain, right? So one thing we can think about is the length. That's sort of obvious on an alpha helix. The size is also not so difficult to imagine. You can imagine an alpha helix composed solely of glycines, which has nothing but protons sticking off of it, or alanines that would have only methyl groups sticking out of it. Whereas a TMD composed of phenylalanines would be very large comparatively because it would have, you know, these large phenols hanging off the side. And the relative, so the way that we define the size is basically by surface area, and it can be measured in a variety of ways, but actually it's pretty easy to predict just straight from a sequence the way that I just talked you through it. And we see for about 100 or 120 now of these constructs that we've created, we see this pretty striking correlation between the total surface area of the transmembrane domain and its raft affinity, with relatively skinny TMDs, those composed largely of glycines and alanines, seemingly partitioning much better to the ordered phase than those which are relatively fat and bulky. And this we discovered after making about 10 different hypotheses that all turned out to be wrong. So it was kind of a correlation that we discovered afterwards. But when we did, it actually made perfect sense when we started thinking about the physical behavior of a transmembrane peptide in a fluid bilayer as basically a solute in a fluid solvent. Anytime you have a situation like that, something dissolved in a fluid, you have an interface. And that interface comes with an energy penalty, right? It costs some amount of energy to push something into a fluid. So that's all quite trivial, but in these phase-separated membranes, any given peptide has the potential to choose between two coexisting fluids. And so there's two different surface tensions that could be available to it. And the larger a peptide is, the more it is likely to be excluded from this tightly packed membrane region where all the lipids are preferentially interacting with one another and are trying to exclude things out of it. And so this cost for pushing proteins into this more tightly packed region basically scales by the total size of the interface, the surface area. And you know, so this was a model that we constructed based on this fit, and to test it directly, we constructed a series of transmembrane domains that were fully synthetic, had nothing left of lat, composed solely of leucines and alanines, whose abundance was just tuned in order to get smaller and smaller and smaller in terms of how fat these alpha helices were. And when we got down to the skinniest alpha helices, we were able to make ones that did prefer the ordered environment, and then the fattest ones with all leucines were excluded, as I showed earlier. And so where we're going with this work is to create what I hope to be a really comprehensive and a predictive model that we don't have to measure raft affinity for every single protein that's out there. We can predict it based on simple physical parameters like the length of the transmembrane domain and how fuzzy it is. And I didn't mention it, but we also think this post-translational palmitylation is a really important component of this model. And so we're far from having this comprehensive predictive model, but so the model we constructed so far certainly does very well when we compare our observations to the predictions from the model. There's clearly outliers and there's clearly much to do, but within this kind of group of constructs, we get pretty good agreement between observation and prediction. So that's cool. We were quite happy with that. But what's really exciting is that the parameters in this model are either fit parameters, like this surface tension difference, or they're things that we can read directly off the sequence of a protein, right? You can just go straight to the database, count how many hydrophobic amino acids a given protein has, and plug it in here. That's the length of the transmembrane domain. Or count up the surface area by counting how many alanines or phenylalanines and plug that number in here and generate a prediction for raft affinity of any protein. And so that's what we've done here. These are our predictions for the raft affinity of all single pass human plasma membrane proteins. And you get this kind of funky looking distribution, which doesn't mean a whole lot. But to test at least some of these predictions, we picked a couple of proteins that were near the seed that we used to build this model. So these three, and then two, which we expected to be very non-raft based on our prediction. And for those five, the model did very well in the sense that the ones that we predicted should behave like LAT did, in that they enriched in the more ordered environment. And the ones that we predicted would be very non-raft were indeed completely excluded from the more ordered environment. So there's still much to do here, but we're excited in where this direction is going. But now I come back to the cell biology that started all of this work, right? So now I claim that we can predict raft affinity from the parameters of the transmembrane domain, physical parameters of the transmembrane domain. But you might be asking, okay, what does this have to do with cell biology? And what I think it does allow us to do is these transmembrane domains are really great tools to somehow visualize the consequences of raft affinity in vivo. And I'll just give you an example here. So the raft hypothesis was originally kind of put forth to explain some aspects of trafficking in a cell. The idea is that we know that the plasma membrane is enriched in things like cholesterol and saturated lipids, glycolipids, certain specific proteins, which led to the hypothesis that there's some sort of sorting that happens in some secretory organelle, like the trans-Golgi network, and that all of the components that belong in the plasma membrane could be packaged together into a trafficking organelle by their mutual preference for an ordered membrane environment. So that would mean that sterols and sphingolipids organize together with certain proteins, and then some mechanism would be used to extract that entire domain for delivery to the plasma membrane. Something like an Amazon pantry for sorting in the cell. And we realized that these TMDs are really a beautiful way to test this model because we have this whole panel of proteins, which are not really proteins at all. They're just transmembrane peptides. They don't interact with any cellular machinery. Instead, they basically mark ordered and disordered domains with this RFP fluorophore. And so then the prediction from this model might be that a raft preferring transmembrane domain would traffic to the plasma membrane, whereas a raft excluded transmembrane domain would not be able to take this path to the plasma membrane and be trafficked differently. And well, that's exactly what we observed. And that now comes back to this data that I showed you at the very beginning, where this isolated transmembrane peptide from LAT traffics to the plasma membrane, whereas this mutant with 6 amino acids removed that doesn't partition as well to the ordered membrane domains also doesn't arrive at the plasma membrane, or rather, not really, right? Accumulates in these intracellular compartments. And this is true not just for this variant, but really across the whole range of constructs that I've showed you so far. We see this clear correlation that if these constructs, some of these like even alanine leucine constructs that have nothing left of lat, if they partition to ordered domains in these GPMVs, they also get to the plasma membrane as well as the wild type lat. However, if we mutate them such that they lose raft affinity in any way, either by making them shorter, by making them all leucines, by mutating pulpitylation sites, in all cases, we lose plasma membrane localization. And it's true not just for lat, but for these other proteins that I showed you earlier. We made truncation mutations of those, and we see that they also accumulate in these intracellular puncta, just like lat. So it seems to be a general phenomenon. And if you're interested in trafficking, you might be asking, well, what are these dots? What are these puncta? And they are degradatory organelles of the endocytic system. So they're late endosomes, here marked by LBPA, and they're lysosomes, here marked by LAMP1. So RAF partitioning proteins get to the plasma membrane. Non-RAF mutants accumulate in lysosomes. And again, also for these other three proteins, very clear accumulation in lysosomes. And from this, we've basically built an understanding, which is very much a model at this point, but I think we have a good bit of data to support this model, that sorting is happening at some endosomal compartment, and a variety of recycling pathways are essential to maintain the homeostasis of the plasma membrane, because the entire plasma membrane sort of cycles through the endocytic system, I don't know, within the hour for sure, but maybe even faster than that. So all proteins need a way to come back to the plasma membrane. One way is sort of a variety of well-characterized sorting motifs that depend on binding to adapter proteins and then motor proteins, excision proteins, etc. But our transmembrane domains have no way to couple into that machinery. They don't interact with any cytoplasmic machinery because they don't have any cytoplasmic amino acids. The way that they recycle and they traffic through the cell is through their ability to partition into a domain that's already going there, and basically hopping on the train that's already going. And I'll wrap up the story by telling you that we're going after the molecular mediators of this recycling pathway. We did a big screen with these same RAFT and non-RAFT transmembrane domains to figure out what knockdowns will lead to their accumulation inside the cell instead of at the plasma membrane. And just very quickly, we fished out these two sister proteins of the same family, RAB3, RAB3A and RAB3B. And that made us really excited because, just very quickly, RABs are really central organizers of subcellular traffic and identifiers of particular organelles. And you won't see RAB3 on this kind of essential cartoon that shows the trafficking from early to late to lysosomes. And we think it belongs here because we find that when we knock down all four isoforms of RAB3, we get massive accumulation of this RAF transmembrane domain in endosomes. And this is specific for the RAF TMD because the non-RAF TMD is unaffected. And so then just very quickly finishing this up, we think that this sorting of RAF proteins is happening at late endosomes because these dots that show accumulations of RAF TMD upon knockdown of all four RAB3 components are late endosomes. They're RAB7 positive late endosomes. And we can actually see these structures by super resolution that are both RAB3 and RAB7 positive and seem to be maybe really generous, handing off this RAF transmembrane domain from one to the other. So this is a cartoon that we're still working on trying to prove, but basically the idea would be that there's a route back to the plasma membrane for endocyte post proteins that requires that these proteins essentially have nothing in common but their ability to partition into an ordered membrane domain. Okay, so in the last 15 or 20 minutes or so, I'm going to tell you a completely different story. Maybe if anybody has any burning questions right away, I'll be happy to address those, or I'll be happy to move on and take questions at the end. Okay, I'll keep going. Okay, so if you don't care about rafts and you don't care about transmembrane domains, you can check back in, because maybe you'll care about this, because it has nothing to do with any of that. The second part of my story is about membrane asymmetry, particularly the molecular and structural asymmetry of the mammalian plasma membrane. So none of this will be news to any of you, so I'll go very quickly. But what we mean by asymmetry is that the outer and inner half, the two leaflets of the plasma membrane bilayer, have different compositions. And this is sort of a fact that's been known for a very long time. The outer leaflet is composed largely of neutral or zwitterionic lipids like sphingomyelin and phosphocholine, whereas the inner leaflet has sort of some lipids are exclusively in the inner leaflet, specifically charged lipids like phosphatidylserine and phosphatidylinositol. And this asymmetry has been implicated in dozens of cellular and physiological processes, although I'm not going to go through any of them because I'm not really convinced that any of them are about asymmetry per se. They're almost always about the exposure of this particular lipid that normally belongs on the inner leaflet, but sometimes gets put on the outer leaflet. I think it's still not entirely clear why the cell expends quite so much energy to sort all of their lipid components against their kind of concentration gradient wishes, and puts a lot of effort into continuing to maintain that asymmetry. But just to quickly come back to where this notion of compositional asymmetry across the bilayer came from. And it came from a very simple set of experiments almost 50 years ago now by some very clever Dutch people. They isolated human red blood cells. Red blood cells only have a plasma membrane. That makes them very convenient for these studies, and they're also an abundant source of cells. And then they applied a variety of lipid cleaving enzymes, so lipases like phospholipase A, phospholipase C, sphingomyelinase, to clip these head groups one at a time. You know, for example, sphingomyelinase here, cutting off these red head groups, and then analyzing what's remaining. And obviously, by implication, what's remaining must be on this inner leaflet, which is inaccessible to this extracellular enzyme. And what was cut must be what was on the outer leaflet. And they did this for one lipid at a time, and that's how cartoons like this were generated. And all of these cartoons that you've seen in various colors and shapes throughout the literature come from this one paper. And even that was a cartoon itself. And the way you know that is if you look at the figure legend, you'll see that this is a cartoon, and it has no error bars because it's a cartoon. And so we thought maybe it'd be useful to revisit this very old canonical data, if only because now we know that there are not 4 lipids in a plasma membrane, but really hundreds. And so to ask how all of those are distributed between the bilayer leaflets. And so we basically took the exact same experimental approach. We even used the very same enzymes from these original paper. And the only thing we did different is dump the lipids into a lipidomics machine instead of TLC, which was much harder and not quantitative. So when we did that, you can generate sort of cool looking lipidomic fingerprints like this, but it's pretty hard to get any kind of meaningful insight from this. But we can quantify all these different species to very high accuracy. And then sort of manually, I combine self-similar species because it's very hard to sort of work with hundreds of different lipids. And I think we can summarize the composition of the outer plasma membrane leaflet with these 6 structurally different lipids The inner leaflet is a bit more complex, but still 10 or 11 or 12 can really capture all of the relevant diversity and abundances of all the different relevant lipid classes. So if you're doing a model membrane study for, say, protein structure function stuff, you could do worse than these compositions. So what did we learn? Well, the first thing that we learned, I think, is that Dutch people are very good at biochemistry, and we shouldn't underestimate them or mess with them, because at least looking at the head group class level, they basically got everything right. These were our distributions of the different head group classes, PCs, PEs, sphingomyelins. These were theirs. Don't bother looking, they're kind of annoyingly similar. And so my face when we finally made this comparison after 2 years and $20,000 worth of lipidomics was very annoyed. But of course, we have more information than they did, because those old methods made it very difficult to know stuff about what was going on in the hydrophobic acyl chains. Head groups are easy, acyl chains are hard. For mass spec, they're both easy. And here we did learn some new things, I think. So namely, we observed that the number of double bonds, so the number of unsaturations in outer leaflet lipids, was very different from inner leaflet lipids. And what you're seeing here in these distributions is the number of double bonds per lipid for outer versus inner leaflet. And what we saw is that the majority, or rather the plurality, of outer leaflet lipids are completely saturated, right? So no double bonds at all. And a pretty significant majority have no more than one double bond, as illustrated there. In contrast, the significant majority of inner leaflet lipids were polyunsaturated, 4, 5, 6, or even more double bonds per lipid, such that the average lipid was almost twice as unsaturated on the inside versus the outer leaflet. And coming back to the membrane biophysics world, for us, this led to the inevitable question: if composition determines membrane physical properties, what are the physical properties of a membrane where the compositions of the two leaflets are so dramatically different? Specifically, a leaflet that would be rich in saturated lipids, like we found the outer leaflet is, would be expected to be very tightly packed, relatively immobile, and not so fluid, and vice versa for an inner leaflet that's the opposite. So to test this idea, we first did some computational simulations. So we constructed these atomistic simulations of an outer plasma membrane leaflet and an inner plasma membrane leaflet, represented by those pie charts that I showed you earlier, and ran some very long and apparently arduous computational experiments. They were easy for me because I just got the data. And our predictions basically turned out to be what we would have trivially predicted, that the outer leaflet is indeed more ordered, more tightly packed, less diffusive than the inner leaflet, right? So this is the diffusion coefficient of a given phospholipid, and you see that an outer leaflet lipid diffuses half as fast as an inner leaflet lipid. And the same is true for lipid packing. The same is true for hydrophobic defects. So there was kind of more hydrophobic defects on the inner leaflet where the lipids weren't packed as tightly. So that was essentially our prediction, and this was essentially the computational result, that the inner leaflet was more disordered, more fluid, with more holes between the lipids than the outer leaflet. So then we wanted to test this idea experimentally, basically, and to do that, we needed a reporter, some way that would measure membrane properties, but do them in a leaflet-specific way, one leaflet at a time. So the first part of that is easy. There's tons of fluorescent molecules that can report on membrane physical properties, like packing. A famous one is called Lordan, but we didn't use that one. We used this other one called Di4. Here's its structure. It has a variety of photophysical properties that can report on membrane packing, for example. And one of these is the fluorescence emission lifetime. And you can see that here, that this dye in an ordered phase emits much slower, about half or twice as slow as the same dye in a disordered phase. And we can even image this in a phase-separated membrane. So now this is the same dye reporting two very different lifetimes, one that's relatively slow and one that's relatively fast in a disordered environment. Okay, so FLIM can be used to report on membrane physical properties, but how do we do this one leaflet at a time in a living cell? Well, that's why we use this dye called dye 4. It has this kind of quasi-head group and quasi-tail. The tail intercalates into membranes, and this quasi-head group is highly charged, which pretty much absolutely prevents it from flipping across the bilayer midplane. And we validated that through a series of controls that I don't really have time to show you. But which means that getting this dye, getting this reporter of membrane properties into the outer leaflet is really easy. We just, toss it into the cell supernatant, and it incorporates by itself, and there reports on the outer leaflet. But the inner leaflet is a little bit harder, but not that much harder. It turns out we can take this very same dye and simply microinject it into the cytoplasm, whereupon it incorporates into the cytoplasmic leaflet of the plasma membrane, and also all of the internal membranes that aren't on this cartoon. So now, again, we have the exact same reporter, but in one leaflet at a time. And we can compare what it reports. So this is an example experiment. This is a mammalian mast cell microinjected with this dye called dye 4. And what you're seeing encoded in the color is the emission lifetime. So this is a FLIM image, and this is the emission lifetime of dye 4. And the cooler colors represent shorter lifetimes, which represent less packed membrane. And so what you're seeing in the cell is that the plasma membrane looks to be a warmer color, kind of green, relative to these blue internal membranes. And that's to be expected. We kind of expected to find that the plasma membrane is more tightly packed than internal membranes because it's more rich than cholesterol, more rich than sphingolipids. Maybe it's non-trivial to point out that we think that this is reporting only the cytoplasmic leaflet. So the cytoplasmic leaflet of the plasma membrane is more tightly packed than the cytoplasmic leaflets of all of these internal membranes, presumably the ER and endosome. But now we can take this very same cell and add the same reporter, dye 4, to the outside, right? So now the plasma membrane is stained from the inside by microinjection and from the outside by just adding it to the supernatant. And we can see that while the internal membranes don't change color, which makes sense, the dye isn't getting to them, the plasma membrane does change color. It gets to be a much warmer color from this kind of green to this orange, suggesting that this newly added dye that's now in the outer leaflet is in a much more tightly packed and ordered environment than the one that was in the inner leaflet. And then we can take some new cells, stain them from the beginning. These now only have the dye in the outer leaflet. And again, we see a shift towards higher lifetimes and therefore more tightly packed membranes. This essentially confirms the computational and sort of theoretical hypothesis that the outer leaflet of the plasma membrane is more tightly packed than the inner leaflet of the plasma membrane in these living mammalian cells. And just for kind of a point of comparison, if you're into the model membrane world, this is a pure liquid ordered phase from a synthetic model membrane, and this is a pure disordered phase from a synthetic model membrane. And you can see that the outer plasma membrane leaflet is almost as packed as a synthetic liquid ordered phase. And the inner leaflet is kind of intermediate between these two. Okay, so that's the plasma membrane, and we realized that we can also do this in internal membranes by essentially staining the cell and then letting the dye endocytose. And when we do that, I'll just go very quickly through this data. We see that endosomes are basically the same color as the plasma membrane when we stain from the outside, whereas when we microinject, they look like the same color as internal membranes. So we think that this asymmetry, this biophysical asymmetry that we measured at the plasma membrane persists after endocytosis up to an hour or so. Okay, And I just want to finish with this thing that I think is really important. So in the first part of the talk, I showed you that proteins can sense their membrane environment, and that skinny proteins preferentially partition to more tightly packed membrane environments, and kind of fatter, bulkier proteins are excluded from this tightly packed zone. But that was all, at least in the cartoons, it was all in a symmetric membrane like this. But now we think that mammalian plasma membranes and some endosomes are asymmetric, with a highly tightly packed outer leaflet and a loosely packed inner leaflet. So how would kind of proteins feel about living in such an anisotropic environment? Well, we can go back to these bioinformatic experiments and ask, does the outer half of a transmembrane domain, is the outer half different than the inner half? So we basically divide these TMDs in half, and we compare the surface area that lives in the outer leaflet to the surface area in the inner leaflet. If you have a symmetric barrel, this asymmetry parameter would be 1, right? Same area here and here. But that's not what we see. So these are the asymmetries in transmembrane domain structures for all human single-pass plasma membrane proteins. And you can see that there's a clear bias that TMDs tend to be asymmetric in the same way that seems to conform to the biophysical asymmetry that we've measured, with a relatively skinny outer half and a relatively fat inner half. And this is true not just for plasma membrane proteins, but also for endosomes. But crucially, it's not true for earlier biosynthetic organelles, ER and Golgi, where the TMDs do appear to be on average symmetric. And so we might predict that there is no biophysical asymmetry in these organelles. And very finally, these are the distributions. Well, this is the distribution I just showed you for mammalian plasma membrane proteins with a clear bias towards this asymmetry. And these are the plasma membrane proteins from the kind of other major eukaryotic clades. And as I hope you would agree, we see the same asymmetry in those single-pass plasma membrane proteins, which to me at least suggests that the biophysical asymmetry that we directly measured in cultured mammalian plasma membranes potentially has been conserved throughout eukaryota, at least as reported by the structural asymmetry of the single-pass transmembrane domains there. And so just to very quickly wrap up, the outer leaflet is mostly saturated, inner leaflet is mostly polyunsaturated, and that leads to a tighter packing in the outer plasma membrane leaflet, and that difference is reflected in asymmetry of transmembrane domain structures. And that allows us to infer that this structural asymmetry, which we, I think, directly observe, and then infer that the biophysical asymmetry persists throughout eukaryotes. And I'll stop here, and I'll thank the brilliant group of people that I'm lucky to work with. Especially, I have to highlight my partner in science and in life, Candace, who's an equal partner in all of our ventures, and Joseph, who did most of that asymmetry stuff, and Barbara, who did most of the first part on the transmembrane domains. And I thank you for your attention, and I'd be excited to take questions.
Okay, that was really, really interesting. Yeah, and very sort of from the first principles, which is very exciting.
The idea.
Oh yeah, that's very very insightful. So, any questions? Anybody want to discuss? Yeah, here start to have questions. So the questions here come in Q&A. So Elizabeth, so let me give Elizabeth. Elizabeth. Okay, here there's several actually scans. So Elizabeth, you can talk. Yeah, I think you're muted.
Okay, cool. Can you hear me?
Yep.
Yep.
Okay, cool. Well, that's really cool that you can predict the raft partitioning by the length of the transmembrane domain, the size, and the lipid modification. For the single pass, I'm wondering about if you looked at some multi-pass transmembrane proteins and if you see some consensus among those different transmembrane domains, or they are different.
It's a really great question. We looked at it a little bit. So our model would sort of predict that multipass proteins would be pretty bad for raft affinity because they're bulky. Even if they're long, they're inherently large, and large was bad in our model for raft affinity. And so we've measured something like 30 multipass proteins and just kind of pick and choosing randomly constructs that we could get our hands on. And so far, out of those 30, some 27 didn't partition to the raft phase at all, which is consistent with this model. But 3 did, and we have no idea why those 3 and what commonalities they seem to share. I can tell you what they are. I'm happy it's actually already published, but so far we really have no idea, and it's a kind of a big problem that I don't know how to even access, because with a single transmembrane domain, already there's a lot of possible variants. With multipass, you have to worry about the helix-helix interface versus the helix-lipid interface. So it's a really essential question that I don't have a good answer for.
Thank you. Can I ask another question?
Sure.
Sure.
So when you did the deletion analysis, like deleting 6 amino acids, and look at the partitioning in the plasma membrane versus the intracellular membranes, if you delete the 6 amino acids from the top half versus the outer versus the inner, do you see a difference?
Yes, we did exactly that to address that exact question. And I would say that we saw some differences that I didn't bother focusing on because I wasn't thinking about this asymmetry question at the time. But now it might be interesting to revisit. So generally, the trend was always that the shorter, the less raft-defined. And I think that's definitely true, but some mutations had bigger effects than others. And so it's making me think that we should revisit that from the asymmetry viewpoint.
Great, thanks.
Okay, Jen had some questions also. Jen?
Unmute. Can you hear me?
Yep.
Okay, great. That was a very interesting talk. I think you went over this part kind of fast. I'm wondering about the screening, like you actually did the screen to look for this RAF recycling pathway.
Yeah.
So so do you see that constantly there is this RAF recycling pathway, and how is, because I kind of saw there's ARF6.
Yeah.
So is that the same as the ARF6 recycling pathway?
Yeah.
Or, like some other thing like retromer, other thing. Can you comment on that?
So the specific route for this raft transmembrane domain recycling, we don't quite know, right? So I did go very fast there. So I'll just very quickly try to recapitulate the idea. We think of these transmembrane domains that partition preferentially to one or the other phase as kind of reporters of that phase, right? Because what they don't do is interact with any proteins. They're just sort of passive peptides that are partitioning in the cell. And so we think where they go, that's where rafts are going. And so when we see an accumulation of those TMDs in the cell, we think that rafts aren't being recycled properly, especially if it's, specific for those TMDs, but not for others. And exactly as you said, when we knock down ARF6 or when we knock down these RAB3s, that's where we see large-scale accumulation of specifically the raft transmembrane domains in late endosome.
Okay, so why late endosome? That seems to be a very, very deep in the in the endosomal pathway. Yeah. Do you have any insight? Is that really what other people studying the ARF? Yeah.
Yeah. So it's not inconceivable that there is sorting from the late endosome. There have been several other proteins that have been shown to do that, and several processes that by which that can happen. And I think you mentioned one, the retromer and RAB9. We probably think that that's ultimately this raft recycling pathway is maybe going through the TGN first, or going through some retromer machinery and then getting sorted again at the TGN. So this sort of idea that it's directly to the plasma membrane, we have no evidence for, and that's for the future. But definitely that there's an arrow for proteins whose commonality isn't say, an adapter protein binding motif, but their ability to partition to these ordered domains. And this is how they get sorted in the cell after endocytosis.
Interesting. Thank you.
Thanks for your question.
So we have a question from the chat. So Weipeng is asking, in the process of endocytosis, the outside of plasma membrane becomes inside of the endosome. So what happens with the components of, saturated and unsaturated lipids on the leaflets?
Yeah, sorry, I went very fast there. That is an important thing that is, usually important to say, right? Exactly as Yiping points out, the luminal leaflet of those endosomes that I showed was just the outer leaflet of the plasma membrane. And no, they don't flip. We find that the luminal leaflet of the endosome is the more ordered one, the more tightly packed one. So I don't know anything about the compositional asymmetry, which lipids are on which side, but I think biophysically we can say that they look similar to the plasma membrane, which sort of makes sense, right? Maybe that you don't get massive remixing of the lipids after endocytosis. They generally look like the plasma membrane in that the luminal leaflet is more tightly packed and the cytoplasmic leaflet, which is still the cytoplasmic leaflet, is more loosely packed.
Very cool. So I had a question. So you focus on plasma membrane mostly, but then when you did these experiments with injection of the dye into the cell, it looks like in the ER, it also wasn't, homogeneous distribution. In the textbook, you usually talk about ER as a sort of very homogeneous sort of membrane because cholesterol levels are low. But from the dye data, it looked like it wasn't really as homogeneous, right? Can you comment on that?
Yeah, it's a really good question. I mean, the one thing I would say is in those microinjection experiments, we're really getting all the membranes. So, we wouldn't be able to tell a ER domain from lipid droplets sitting on the ER or a lysosome sitting next to the ER, right? So some of those color differences may be that they're different organelles. That said, I think you're exactly right that this approach and similar ones can be used to map the differences in physical properties between organelles, and that's actually already been done, and it's consistent with what you might predict. The ER is the most fluid, Golgi becomes less so, plasma membrane is the most packed. But within organelles as well, and it just becomes a resolution game. But I actually think you're right. We have some evidence from trafficking data and some from imaging that suggests that the ER is maybe not homogeneous.
Very cool. Any other questions? Anybody? Last call. Okay, thank you very much, Ilya. So I'm sure people have a lot more questions. They will write you and contact you. And I think we are now switching off this Zoom.